Hello, my name is JoAnna Holm, and I am a postdoctoral fellow at the Institute for Genome Sciences in the University of Maryland School of Medicine. Today I'm going to share with you my experience shining light on an elusive bug through a comparative metagenome-assembled genome analysis of Candidatus Lachnocurva vaginae, formerly known as Bacterial Vaginosis Associated Bacterium 1. The vaginal microbiome is a complex and dynamic host-associated community found in close association with the vaginal epithelia. Hypothesized roles of the microbiota are displayed here. Starting at the bottom left of the slide, estrogen induces the production of glycogen in the vaginal epithelial cells, and it is this glycogen which is a nutrient source for various species of gram-positive lactobacillus. The presence of lactobacillus can, in turn, impact the host's reproductive health through various mechanisms, including defense against pathogens, and it's associated with positive health outcomes, such as, in this example, a healthy pregnancy brought to term. Another commonly observed vaginal community is broadly referred to as community state type 4, or CST4, which does not contain lactobacillus species, but rather a diverse array of obligately anaerobic bacteria. CST4 is associated with an unfortunately common condition referred to as bacterial vaginosis, or BV. Women with BV experience troublesome symptoms, including vaginal discharge, itching, burning, and odor, which can impact a woman's self-esteem and sexual health. In addition, BV is a risk factor for other negative sequelae, which include preterm labor and acquisition of sexually transmitted infections, including HIV, and can contribute to a huge economic burden globally. The vaginal microbiota is commonly surveyed just like other environments using the 16S ribosomal RNA gene sequence. In this heat map, each column is a sample, and each row represents the relative abundance of that taxon measured in each sample, where 0 is represented by beige and 100% dominance in a sample is represented by red. CST4, in the two shades of blue, is divided into two groups, A and B. CST4A is dominated by Bacterial Vaginosis Associated Bacterium 1, or BVAB1, the bacterium of interest in this study. BVAB1 is the only major vaginal species for which a cultured isolate has not been obtained, and until this study, there were no reference genomes available. BVAB1 was only known to exist through the presence of the 16S rRNA gene sequence between the 3rd and 4th variable regions. The molecular identification of BVAB1 was reported in the New England Journal of Medicine in 2007. The microscopic image in the top right is a fluorescent in situ hybridization experiment in which BVAB1 was tagged with green fluorescent stain. You can see the morphology of those bugs is a curved rod. Since then, BVAB1 has been the topic of numerous studies, especially in the context of bacterial vaginosis and other negative health outcomes. For the purposes of my research, there was a critical gap of knowledge in that BVAB1 could not be identified in metagenomic data. With 800 metagenomes on their way to me so that I could study the effect of the vaginal microbiome on incident sexually transmitted infections, I needed to know which genes belonged to BVAB1. And so the primary goal of this study was to obtain a full genome of as yet uncultivated BVAB1. With the limited knowledge that we glean from the short 16S ribosomal RNA sequence, that it belongs within the bacterial order Clostridiales and is abundant in some vaginal samples. There is the additional limitation that vaginal metagenomic sequence data, just like other host-associated metagenome data, is predominantly composed of host sequence data. So to begin this analysis, I chose vaginal samples from my study, as well as the Human Microbiome Project, which were dominated by BVAB1 according to 16S ribosomal RNA gene sequences. The 7 samples denoted here came from women 6 of which were clinically diagnosed with bacterial vaginosis. Not all of the women reported symptoms though, and this is denoted as asymptomatic or ASYM in the BV status column. Gram stains from each of these samples in the bottom of the slide shows a predominance of the gram-positive curved rods, presumed to be BVAB1. 6 of these samples already had short-read metagenomic data from the Illumina HiSeq 4000 platform. The 7th sample was from the Human Microbiome Project and had enough high-quality DNA to allow for metagenomic sequencing on the PacBio Sequel II platform. Here's a table where the metagenomic library preparation and sequencing parameters are summarized. Samples sequenced on the HiSeq Illumina platform were sourced from cervicovaginal lavage fluid. The DNA was extracted using the QIAGEN MAG-Attract microbial DNA kit, and libraries were prepped with the Kappa Hyper Plus kit. The libraries were sequenced on the Illumina HiSeq 4000 platform, and human reads were removed using BM Tagger and quality filtered using Trim-O-Matic. Finally, assembly was performed using Spades. The sample for Sequel II sequencing was sourced from a Copan eSwab, from which DNA was extracted using the Lucigen Master Pure DNA purification kit. The library was produced using the PacBio SmartBell template prep kit and sequenced on the Sequel II platform with 4 other libraries. Human read removal was performed using PB-Align, and the assembly was completed using Canoe version 1.7 using the PacBio Raw option and other default parameters. I want to take a moment to clarify the assembly of the Sequel II produced reads, as at the time, there was no procedure in place for this, for sequences coming off of the Sequel II platform. So following a lot of troubleshooting, we obtained the best assembly by inputting subreads to the Canoe assembly program, which has its own algorithm for calling consensus reads. So here are some assembly details for each sample. Using the PacBio Sequel II platform and the Canoe assembly program as specified, we obtained a single circularized contig identified as BVAB1. This contig contained 1,434 coding sequences at a total length of 1.64 million base pairs and an average of 124x coverage. The assembly also included 4 complete ribosomal RNA operons. Comparatively, using the Illumina HiSeq 4000 platform and the Spades assembly program, the BVAB1 genome was spread across as few as 26 contigs to as many as 152 contigs. And while mean coverage was high, about 2256x, there were many regions found on the Sequel II based assembly that were absent from the HiSeq based assemblies. To provide an equal comparison, the UAB071 genome was also sequenced on the Illumina HiSeq platform and assembled as specified previously. In this figure, which is from the program NUCMER, the Sequel II assembly, which is the single contig, is on the y-axis, and the HiSeq assembly, which has many contigs, is on the x-axis. So you can see that while there's a lot of overlap of the various regions of the genome, it's not entirely covered. And even more importantly, we were able to get gene order using the Sequel II based single contig. So what did we learn from this analysis about BVAB1? Excuse me. Well, all of the results from this can be found in a publication reported earlier this year, but I am going to summarize briefly some of the major findings for you here. First, as stated earlier, the metagenome-assembled genome contained 4 complete RNA operons. And so 4 complete rRNA operons. So using the newly obtained full-length 16S ribosomal RNA gene, we were able to phylogenetically place BVAB1 within the family Lachnospiraceae, and further recommend its placement into a novel genus, Lachnocurva. This required a renaming of the actual species, and so we proposed Candidatus Lachnocurva vaginae. Lachno indicates its presence within the family Lachnospiraceae. Curva represents the curved rod morphology of the species. And vaginae emphasizes the originally sourced location of the species. Here is a visual representation of some of the major genomic findings of this species. First of all of the genes required for flagellar assembly were found in the genome. This is fascinating because no vaginal species has ever been found to be motile. In addition, genes for chemotaxis were observed, which could stimulate the organisms to move towards nutrients or away from toxins. We also observed a transporter which imports betaine, carnitine, and choline, as well as two important genes known as CUT-C and CUT-D, which have been previously described in gut bacteria. Well, this group of genes is important because the CUT genes can turn choline into trimethylamine, or TMA, the oxidized form of which produces a fishy odor. So while the source of this oxidation remains unknown, this is evidence that Candidatus Lachnocurva vaginae may contribute to the notable fishy odor symptom of bacterial vaginosis. We also observed multiple genes involved with defense mechanisms, which include the evasion of antibiotics and the production and export of bacteriocins. Finally, we identified genes which provided clues into the main nutrient sources used by Candidatus lachnocurva. Namely, this includes mannose, fructose, and L-ascorbate. And this information will be important for future attempts to cultivate this organism. This is a visual representation of the 1.6 million base pair metagenome-assembled genome of Candidatus lachnocurva vaginae. While we already talked about some really exciting genomic features, what we see here is that there's actually quite a bit of potential for mobile elements. So the orange rectangles around the outside of this figure denote sites of genomic islands or mobile elements. So further sequencing of this species, which can be enhanced using this circularized metagenome-assembled genome, can expand the pangenome of this species, and this will give us further insights and clues into the amounts of potential horizontal gene transfer occurring, which can contribute to the pathogenesis of this organism. For me, the future impact of this study was that I could finally define genes from the metagenomes that belong to Candidatus lachnocurva vaginae. One of my primary goals has been to define and characterize metagenomic community state types of the vaginal microbiome. And as you can see here, we have MGCSTs 16 through 18 in the tealish greenish color. And these are all dominated by various metagenomic subspecies of Candidatus lachnocurva vaginae, which was directly informed through this genomic analysis. My hope and goal is that my research will produce novel ways of reducing bacterial vaginosis and other negative health outcomes for women and their reproductive health. With that, I'd like to thank you for your attention today. I'd also like to thank PacBio for the invitation to speak to you and share my research. I'd also like to thank the Ruth L. Kirstein National Research Service Award through the NIH-NIAID for funding, and the University of Maryland Genomics Resource Center for the opportunity to sequence this elusive organism. I'd be happy to take questions in the following live session.
Good afternoon, everybody. My name is Nick Cisneros. I'm the Senior Director of the Field Application Scientist team here at PacBio. And with me is Dr. JoAnna Holm, who's a postdoctoral fellow in the Raval lab at the University of Baltimore, Maryland. Please go ahead and answer or type in your questions into the chat box. We'll be happy to get those answered for you as we get moving here. To start us off, there are a few questions already. Since the publication of these findings, have you had success culturing this sample?
No, actually, we, within our lab, we haven't even been able to take time to do it, honestly, because our lab switched over to some COVID testing So there's not been a lot of opportunity to make that effort yet.
Okay. What kind of issues do you think there are in the lack of culturability?
I think it's a lot of nutrition and a lot of requirements. The BVAB1 Candidatus Lachnocurva vaginae wasn't quite so good at making its own amino acids. A lot of the nutrients it appeared, the sugars it appeared to have access to are things that are sort of breakdown products of other things. So I think it's largely due to it's used to living with a lot of other organisms and sort of being a scavenger off of those pieces.
Interesting. So the lack of the presence of those other organisms makes it almost unable to grow without that. That's interesting.
I think so, and I think there's probably a chance it has been produced in a culture before, but maybe folks couldn't recognize it for one reason or another because it's maybe not azenic, right? It's in community with something else, and so once you get to an isolated culture, you probably don't have BVAB1 anymore. But now that we know a lot more, let's hope that can happen soon.
Sure. And there's starting to have some questions start to roll in. Were the 4 16S gene copies identical in sequence? If not, how is diversity in multiple 16S genes for a single organism captured in the databases?
They were not identical. They were a few base pairs different. Can you say the second part of that question again?
Yeah, how is diversity in multiple 16S genes for a single organism captured in the databases?
Oh, so in this case, all of the 16S copies, the whole ribosomal RNA operon would be within the genome. So one could go in and grab those annotated pieces from the genome pretty easily.
Okay.
Perfect.
Yeah. The next question is, did you look at the methylome of this organism?
No, we haven't. This is what you see is what you've gotten so far. We haven't done too much more. What we do have, which I don't have results on yet, is we do have some metatranscriptomic data for metagenomes of samples like this that are dominated by Candidatus Lachnocurva vaginae.
Okay, that's fantastic. And then one other questions. Have signs of, I'm going to mess this up, I already told you, Lachnospiraceae vaginae been flagellated, been confirmed morphologically?
No, and this was something else that being back in the lab would be great to do because there are some flagella-specific stains that one could use on just slides, and we haven't been in there to do it. So it's a great.
Okay, perfect. That seems to be all the questions we have for today.
Great.
So thank you, JoAnna, for your time. That was a fantastic talk, and we look forward to seeing the rest of the results here. It sounds like you got a lot of interesting stuff still yet to come, and we look forward to seeing how that pans out for you.
Thank you. Yeah, we're really excited about a lot of the impacts we can hopefully make in improving women's reproductive health outcomes. So thanks for the opportunity today.
Yeah, thank you, and stay safe out there, and we'll talk to you soon.
You too.
Thank you, everybody, for joining us. Have a good rest of the afternoon.